Hello there, anatomy and physiology folks. Let's talk about the human brain. When we look at the brain, this is kind of what we think of when we sort of picture it in our minds. We see this sort of wrinkly looking thing and this brownish, grayish, yellowish stuff, right? This. Okay, this is our model that we have in the classroom. Um, I want you to understand when you're seeing a brain like this, it's been preserved, so it's not naturally this color. Um, when you look at a brain that's fresh, so to speak, it looks more like this. This is a brain that's been freshly removed from a deceased patient. It looks like this is an autopsy table here. This is the foot of the decedent. This brain is pink and white, and the blood here is in these vessels that are on the surface of the brain, of course. This brain, something here looks a little suspicious to me. Maybe there's some type of cerebrovascular accident here, or maybe some brain bleeding here. And it looks like there was a section of the brain taken here. It looks like this is a frontal section, and this is a transverse section here. Part of the frontal lobe of this brain was removed, perhaps for inspection in and around this area. If you look at this brain, this is anterior, this is posterior. You can see the cerebellum here. And I'll show a video in class, or I'll post a video to the class website that shows how floppy the brain actually is when it's not been preserved and fixative. The brain grows quite a lot in the first few years of life. There's a quite tremendous rate of growth from embryonic development through about the age of 2, and the rate of brain growth slows down a little bit up until about the age of 5, and it still continues to grow and getting bigger through about the age of 10. On or about, your 11th or 12th birthday, your brain is about as big as it's ever going to get. Interestingly enough, the size of the brain is roughly 2% of the body, but the energy requirements is tenfold that, right? 20% of the body's energy is devoted to operations in the brain, which is an indicator that what the brain is doing is really important. And I think that's probably obvious to most of us. The literature varies in indicating to us how big the brain is in terms of number of cells. Your textbook says there's 1 trillion cells. There's some other literature out there that suggests the number is somewhere around 100 billion. Either way, it's quite a large number of cells. It is one of the larger organs of the body, not as massive as your skin or your liver, but it is pretty big relative to the other organs in your body. On average, the mass is about 3 pounds, although that can vary from person to person. There are 4 principal parts of the brain: the brain stem, the diencephalon, the cerebrum, and the cerebellum. Those last 2 parts of the brain listed here, cerebrum and cerebellum, those words are really similar, so make sure that you're careful when you're reading and when you're writing. As you look at the brain like this is a model of the brain. This is a mid-sagittal view of the brain. You're seeing the left half of the brain, if you can picture it right. This would be anterior, this would be posterior, superior is at the top here, inferior is at the bottom. And then the 4 principal parts of the brain are labeled here. This is cerebrum, and then the diencephalon is sort of like the central structure of the brain. The thalamus and the hypothalamus are substructures of the diencephalon. The brain stem includes a structure known as the midbrain, the pons, and the medulla, or medulla oblongata. And then this part back here, which is a separate part, is called the cerebellum. And then, of course, the spinal cord is going to emanate inferiorly. It's actually contiguous with the medulla oblongata. And where the spinal cord begins and where the medulla ends is essentially where the foramen magnum of the skull is. Let's look a little bit more at the brainstem here. Again, the midbrain area, this color green here is a false color, of course. The pons is here in blue, and the medulla oblongata is in orange, right? And you can see relative to where those structures would be over here relative to the entire brain. Let's move on here. The diencephalon, of course, is including the thalamus and then the hypothalamus. There's some other structures here that are indicated. This corpus callosum, this is the structure that really kind of holds, and then some of these commissures. These are the structures that hold the 2 halves of the brain together, and that's where there's contralateral communication between the hemispheres of the cerebrum. talk a little bit more about that. There's other parts here that are labeled that you can pause the video and look at this diagram and study a little bit as you need. The cerebrum is like the part of the brain that you see. When you first are looking at a brain, like the most obvious part is the cerebrum. You see these sort of grooves and these wrinkly sort of bumpy structures in the brain. That's the main portion of the brain is the cerebrum. And this is really where a lot of the sort of executive functioning, if you will, of the brain is occurring, like where the decisions are made for your body to do what, and your brain is processing its sensory information. It's a really important portion of the brain. They're all important, of course. This is looking at the posterior, so this blue part here, and of course your brain is not blue, is the cerebellum. So this part that's this beige yellow color is the cerebrum. And this is another look at the cerebrum and looking at the varying portions of the brain. We have these structures called lobes, which are just the regions of the brain, if you will, and they're generally named like this frontal lobe here and the parietal lobe indicated here. They're named for the bones that those portions of the brain are directly deep to. Notice here that the term sulcus and the term gyrus are indicated here. A sulcus is a groove, like one of these folding-looking inward sort of grooves in the tissue, and a gyrus is sort of the upward fold or the protrusion in between the grooves of brain tissue. This major sulcus right here is known as the central sulcus. It's usually relatively easily identifiable in human brains. And so precentral and postcentral, those terms are relative to the location of this central sulcus. Obviously, this is occipital lobe, and then we have the temporal lobe. Those, again, are named for bones that the regions of the brains are underneath. And then we see the other parts of the brain here in this diagram as well. The cerebellum is this back portion of the brain. And if you take a cut through it sagittally, mid-sagittally, you'll see this interior structure that kind of looks like a tree. And you guys probably know what Arbor Day is, or an arborist. This means tree. So this white matter in between the gray matter kind of looks like a tree. And it's named Arbor Vitae, which literally means tree of life, which is going to be a really important structure here. The cerebellum is largely important for coordinating and perceiving your body movements and body position. Brain's really important, so it has some layers of protection. Of course, there is skin, and there is bone, and of course we have the meninges, right? So we have the dura mater, the arachnoid layer, and the pia mater. The dura mater is the most superficial of those three, and the pia is in direct contact. This material here, this blue stuff, as indicated, this is where the cerebrospinal fluid is. And the brain is essentially floating in that cerebrospinal fluid. When we're looking at these three layers of meninges, you can see here number 1, number 2, number 3. These are the dura mater, the arachnoid layer or mater, and the pia mater specifically. This is going to be where cerebrospinal fluid is flowing. Okay, there's actually 2 layers of dura mater membrane. One's called the endosteal, and one's called the meningeal layer. The arachnoid membrane, you notice, kind of looks webby, like fibrous. So that's why it's kind of looks like a spider web. That's why it's called arachnoid. And then the pia mater is going to be in direct contact and bound to the surface of the neurons that are in the brain here by the glial cells, those astrocytes. And a really important structure here, of course, is there could be blood vessels in here, but the blood doesn't go down into this structure here. But what you don't want in any type of situation is for blood vessels that are in here to be bleeding. If you have a subarachnoid bleed, it's not a good situation. Cerebrospinal fluid is going to be flowing through this area too, and in here, delivering another mechanism for delivering nutrients and oxygen to the cells of the brain. This shows you, the blood-filled dural space, and then this here is going to be cerebrospinal fluid, this lighter blue on this color. Now, cerebrospinal fluid and blood is not blue, of course. It's just false color here on this diagram to make it a little bit more clear for you. The choroid plexus is part of this diencephalon region of the brain here. cells of the choroid plexus are the ones that are actually generating the cerebrospinal fluid and making this stuff, which is essentially, I mean, it's kind of a filtrate of blood. So if you know what a filtrate is, it's stuff that can pass through a filter. And this is the pathway. These arrows here indicate the pathway of the flow of cerebrospinal fluid. And I want you to notice that these ventricles of the brain, here's the third ventricle, this is going to be the fourth ventricle in here. It's not labeled. Oh, it is labeled, I'm sorry. That's going to be contiguous with that central canal in the spinal cord. And there's a pathway through for cerebrospinal fluid to circulate up and around the cerebellum here, and then, of course, following around the cerebrum as well. The ventricles, when you look at them, there are these spaces in the brain. It's kind of hard to visualize where the ventricles are when you're looking at a mid-sagittal section. So you've got this kind of diagram that sort of shows you how the structure of the ventricles is really organized. You can pause the video and look at these labels here and study a little bit more about how the ventricles are labeled, the different regions of the ventricles are labeled. This is what the blood vessel network on the surface of the brain that's sitting within the membrane, so underneath the dura, but not actually diving down into the tissue of the brain itself. It's a very complex and vast network of blood vessels, and generally the blood vessels follow the path of the grooves, that is the sulci or the sulci of the cerebrum. And blood delivery to the brain to make cerebrospinal fluid so that those neurons can be bathed and washed in nutrients and oxygen is really super important, obviously. And when blood flow is compromised for some reason to the brain, if there's a blockage in arteries that deliver blood flow to the brain, we call that a stroke, and part of the brain can die as a result of that. And if the blood vessels rupture, we call that a hemorrhage. If it's a rupture within some of these major arteries that deliver blood in between the grooves or these fissures or sulci, we call it intracerebral hemorrhage. And if the brain tissue gets sort of filled up with blood, if the blood actually leaks out of a ruptured blood vessel and gets into the tissue itself and maybe starts to fill the ventricles, that's not good. Hematoma is the word we describe, word we use to describe when there's a region of tissue that's full of blood that shouldn't be. Okay, that's it for this video. Hopefully that helps you.